Hello there, and welcome to the Sleepy Science Society. Tonight, we're embarking on a journey through one of the most profound discoveries in all of science, a story written in stone, in DNA, in the bones of creatures long vanished, and in the living world all around us. We're going to explore evolution, the grand unifying principle that connects every living thing on Earth in an unbroken chain stretching back billions of years. If you find these gentle explorations of science soothing, I invite you to like and subscribe so more curious minds can find their way to this quiet corner of discovery. But for now, settle in, let your thoughts drift, and allow yourself to wander through deep time with me. Evolution is more than just a scientific concept. It's the story of life itself, a narrative so vast that it encompasses every creature that has ever drawn breath, every plant that has ever reached toward sunlight, every microorganism that has ever divided in the hidden depths of the ocean. At its heart, evolution describes a simple yet elegant truth. Living things change over time, and these changes accumulate across generations, shaping the diversity of life we see today. It's a process so gradual that we rarely notice it in our brief human lifetimes, yet so powerful that it has transformed single cells into the magnificent complexity of redwood forests, coral reefs, and human consciousness itself. The word evolution conjures different images for different people. Some envision the famous progression of figures showing an ape gradually straightening into an upright human. Others think of Darwin's finches on distant islands. Still others picture the spiraling double helix of DNA, that twisted ladder carrying instructions refined across countless generations. All of these images capture fragments of the truth, but evolution is bigger than any single picture. It's a process that operates across scales both vast and minute, from the molecular changes in a single gene to the rise and fall of entire ecosystems across millions of years. Tonight, as you rest and your mind grows quiet, we'll trace the threads of this grand narrative. We'll visit volcanic islands where a naturalist first glimpsed the pattern. We'll descend into ancient rock layers where fossilized bones whisper secrets of vanished worlds. We'll peer into the microscopic realm where DNA reveals family connections between all living things. We'll witness evolution happening in real time, in hospital wards where bacteria evolve resistance, in laboratories where fruit flies change before scientists' eyes, and in wild populations responding to environmental shifts. This is not a story with a single beginning or a final ending. What continues to evolve, continues to change, continues to respond to the pressures and opportunities presented by an ever-shifting world. We are not observers watching from outside this process. We are participants, our own species shaped by the same forces that carved the wings of birds and the roots of trees. Our DNA carries the history of our ancestors, stretching back through early humans, through ancient primates, through mammalian precursors, through fish-like creatures in primordial seas, all the way to the first flickering sparks of life itself. So breathe deeply, let your shoulders relax, and prepare to travel through time. We'll move slowly, pausing to examine the evidence, to understand the mechanisms, to appreciate the beauty of this profound insight into how life works. evolution is patient. It operates on timescales that dwarf human civilizations. And tonight, we have all the time in the world to explore its wonders. Let's begin our journey where the modern understanding of evolution began, on a ship sailing toward distant shores, carrying a young naturalist whose observations would change everything. In 1831, a 22-year-old naturalist named Charles Darwin stepped aboard a ship called the HMS Beagle, beginning a 5-year voyage that would fundamentally reshape humanity's understanding of life on Earth. Darwin had studied to become a clergyman at Cambridge University, but his true passion lay in natural history. He collected beetles, studied geology, and harbored an insatiable curiosity about the natural world. When the opportunity arose to join the Beagle as the ship's naturalist, documenting the flora, fauna, and geology of distant lands, Darwin seized it. The Beagle's mission was to chart the coastlines of South America and survey various islands across the Pacific. For Darwin, it became something far more significant, a floating laboratory where he could observe the stunning diversity of life across different continents and isolated islands. He collected thousands of specimens, meticulously recorded observations in his notebooks, and puzzled over patterns that didn't quite fit the prevailing view that species were unchanging creations, each placed in its environment by divine design. Everywhere Darwin looked, he saw variations. In the rainforests of Brazil, he marveled at the riot of plant and animal life, each species seemingly perfectly adapted to its niche. In the grasslands of Argentina, he unearthed fossilized bones of giant extinct mammals that resembled yet differed from the living animals inhabiting the same regions. These extinct giants posed a troubling question. If species were created perfect and unchanging, why did so many go extinct? And why did the extinct forms bear such uncanny resemblances to living species in the same geographic areas? But it was the Galapagos Islands, a cluster of volcanic rocks straddling the equator off the coast of Ecuador, that would provide Darwin with his most crucial insights. The Galapagos were relatively young islands, geologically speaking, having emerged from the sea through volcanic activity perhaps only a few million years earlier. They were never connected to the mainland, meaning every living thing on these islands had to arrive by sea or air, carried on ocean currents or blown by winds. What Darwin found on these isolated islands was remarkable. Each island had its own distinct forms of life, particularly among the mockingbirds and the finches that would later bear his name. The mockingbirds on different islands were similar, yet each island population had its own characteristics. The finches were even more intriguing. They came in different sizes, with different beak shapes, each seemingly suited to different food sources. Some had thick, powerful beaks perfect for cracking seeds. Others had long, slender beaks, ideal for probing flowers or catching insects. Still, others had beaks adapted for eating cactus fruits, or even using tools to extract grubs from tree bark. Darwin didn't immediately grasp the full significance of what he was seeing. It would take years of reflection, correspondence with other naturalists, and careful examination of his specimens before the pattern became clear. But the seeds of understanding were planted there among those volcanic rocks. If all these finch species had descended from a common ancestor that had somehow reached the islands from the mainland, And if populations on different islands had been isolated from each other, then perhaps the variations between them weren't random. Perhaps they reflected adaptation to different island environments and different available food sources. Back in England, Darwin began working through the implications of his observations. He filled notebook after notebook with thoughts, questions, and tentative theories. He studied the work of domestic animal breeders, noting how they could produce dramatic changes in pigeons, dogs, and livestock through selective breeding. If humans could reshape species through artificial selection, choosing which individuals to breed based on desired traits, could nature do something similar? The key insight came from thinking about population dynamics and the struggle for existence. Darwin had read Thomas Malthus's essay on population, which argued that populations tend to grow faster than their food supply, leading to competition and struggle. Darwin realized that this principle applied not just to humans, but to all living things. Most organisms produce far more offspring than can possibly survive to reproduce. A single fish can lay thousands of eggs. A dandelion produces hundreds of seeds. Yet populations remain relatively stable over time. Most of those eggs, most of those seeds, must die before reproducing. Here was the mechanism. In any population, individuals vary. Some are faster, some stronger, some better camouflaged, some more resistant to disease. These variations are often heritable, passed from parents to offspring. Now, in the struggle for existence, individuals with variations that help them survive and reproduce in their particular environment will, on average, leave more offspring than those without such advantages. Over time, beneficial variations will become more common in the population, while detrimental ones will disappear. Darwin called this process natural selection, drawing an explicit parallel to the artificial selection practiced by breeders. But natural selection operates without conscious choice, without foresight, without any guiding hand. It's simply the inevitable mathematical result of variation, heredity, and differential survival. Given enough time, enough generations, natural selection could transform species, splitting populations into new forms as they adapted to different environments. Darwin spent decades gathering evidence, refining his theory, and filling his notebooks before he felt ready to publish. He might have waited even longer if not for a letter that arrived in 1858 from a young naturalist named Alfred Russel Wallace. Wallace had been conducting his own observations in the Malay archipelago, and he had independently arrived at nearly the same theory of evolution by natural selection. Wallace's letter contained a manuscript outlining his ideas, and he asked Darwin to review it. Darwin was stunned. Years of careful work, and now someone else had reached the same conclusion. With the help of colleagues, Darwin and Wallace agreed to present their ideas jointly to the Linnean Society of London. The papers were read in July 1858, though neither Darwin nor Wallace attended the meeting. The response was muted. The scientific community didn't immediately recognize the revolutionary nature of what they'd heard, but Darwin now knew he needed to publish. In 1859, he released what he called an abstract of his larger work, a book titled On the Origin of Species by Means of Natural Selection, or the preservation of favored races in the struggle for life. The book was an immediate sensation. The first printing sold out on the first day. It sparked fierce debates, passionate defenses, and vehement denunciations. Some embraced Darwin's ideas enthusiastically. Others rejected them outright, finding the notion of humans sharing ancestry with apes deeply troubling. Yet Darwin's argument was powerful because it was built on evidence. He didn't just propose natural selection as an abstract principle. He showed how it explained patterns in domesticated animals, in the geographic distribution of species, in the relationships between living and extinct forms, in the embryological development of different species, and in countless other observations. He demonstrated that species weren't fixed and unchanging. They were malleable, shaped by their environments over immense stretches of time. What Darwin couldn't explain was how inheritance worked. He didn't know about genes or DNA. The mechanism by which traits passed from parents to offspring remained mysterious to him. It would take decades and the work of many other scientists before the picture became complete. But Darwin had given the world a framework for understanding the diversity of life, a framework so powerful and explanatory that it would become the foundation of all modern biology. As you rest tonight, consider the patience and dedication required to develop such revolutionary ideas. Darwin spent years observing, collecting, questioning, and synthesizing. His theory emerged not from a single flash of insight, but from the gradual accumulation of evidence and the slow, careful work of reasoning through its implications. It's a reminder that the greatest scientific discoveries often require time, persistence, and a willingness to follow evidence wherever it leads. Now, let's explore the mechanisms of evolution in greater depth, understanding how the process actually works at the level of populations and genes. Evolution operates through several interconnected mechanisms, but natural selection remains the most important process shaping adaptive change. To understand how natural selection works, we need to grasp three fundamental requirements: variation, heredity, and differential reproduction. These three elements together create the conditions for evolutionary change. First, there must be variation within a population. No two individuals are exactly alike. Even members of the same species differ in countless ways. Some are larger, some smaller. Some have darker coloration, others lighter. Some are faster runners, others more resistant to cold. Some have more acute vision, others better hearing. This variation provides the raw material for natural selection to act upon. Without variation, natural selection has nothing to work with. A population of identical individuals cannot evolve because there are no differences for the environment to favor or disfavor. Where does this variation come from? The ultimate source is mutation, changes in the DNA sequence that occur when genetic material is copied. DNA is a remarkably stable molecule, but it's not perfect. Occasionally, when cells divide and their DNA is replicated, errors creep in. A single nucleotide might be swapped for another. A section of DNA might be duplicated or deleted. These changes are usually random with respect to what would be beneficial for the organism. Mutations don't arise because they're needed. They just happen, spontaneously, driven by chemical processes and occasional copying mistakes. Most mutations have no noticeable effect. They might occur in stretches of DNA that don't code for anything important, or they might cause such subtle changes that they don't significantly impact the organism's survival or reproduction. Some mutations are harmful, disrupting important biological processes. These tend to be quickly eliminated by natural selection because individuals carrying them are less likely to survive and reproduce. But occasionally, very rarely, a mutation proves beneficial. It might make an enzyme work more efficiently, enhance disease resistance, or improve some aspect of the organism's function in its particular environment. Once a beneficial mutation arises, it can spread through a population over generations. This is where heredity comes in. Traits must be heritable, passed from parents to offspring, for evolution to occur. If beneficial variations arose but couldn't be inherited, they would die with the individual and couldn't accumulate over time. Fortunately, the genetic basis of traits ensures that characteristics are passed down. When an organism reproduces, it transmits copies of its DNA to its offspring. If that DNA carries a beneficial mutation, the offspring will inherit it. This brings us to the third requirement: differential reproduction. In nature, not all individuals have equal reproductive success. Some leave many offspring, others few or none. The reasons for this differential success are myriad. An antelope that can run slightly faster might escape predators more often, living longer and producing more fawns over its lifetime. A plant with deeper roots might survive droughts better, setting more seeds than shallow-rooted neighbors. A bird with a slightly more efficient beak shape might gather food more quickly, raising more chicks each season. The key insight is that if these advantageous traits are heritable, if fast-running antelopes tend to have fast-running offspring, if deep-rooted plants produce deep-rooted seedlings, then over generations, the beneficial traits will become more common in the population. This is natural selection in action. It's not about individual organisms evolving during their lifetimes. Rather, it's about the composition of populations shifting over generations. As some genetic variants become more common and others less common or disappear entirely. Natural selection acts on phenotypes, the observable characteristics of organisms, but it ultimately changes genotypes, the genetic makeup. An organism's phenotype is the result of complex interactions between its genes and its environment. Genes provide instructions, but how those instructions are expressed can depend on environmental factors. A plant's height depends on its genes, but also on the availability of water, nutrients, and sunlight. Natural selection favors phenotypes that enhance survival and reproduction in particular environments. And in doing so, it indirectly favors the underlying genetic variants that contribute to those phenotypes. It's important to understand that natural selection is not a force with intention or foresight. It doesn't plan ahead or work toward any goal. It's simply the statistical result of some individuals leaving more descendants than others. If the environment changes, the traits favored by selection will change too. What's adaptive in one environment may be maladaptive in another. The thick fur that helps a mammal survive in Arctic cold would be a liability in tropical heat. Evolution has no direction except the one imposed by immediate environmental conditions and the current genetic variation in the population. Beyond mutation, other processes contribute to genetic variation. Sexual reproduction, practiced by most animals and many plants, shuffles genetic material in ways that create new combinations. When organisms reproduce sexually, they combine genetic material from two parents. The offspring inherits half its genes from each parent, but which half is somewhat random, determined by the particular sperm or egg involved in fertilization. This mixing and matching of genetic material means that siblings can be quite different from each other, each inheriting a unique combination of parental genes. Recombination, which occurs during the formation of sex cells, further increases variation. During meiosis, the process that creates eggs and sperm, chromosomes can exchange segments with each other, creating new combinations of genes that didn't exist in either parent. These processes ensure that even without new mutations, sexual reproduction generates tremendous variation among offspring. Gene flow, the movement of genes between populations, is another source of variation. When individuals from one population migrate to another and breed, they introduce new genetic variants. This can prevent populations from becoming too genetically isolated and can spread beneficial mutations more quickly. On the other hand, genetic drift, random changes in gene frequencies that occur especially in small populations, can cause evolutionary change independent of natural selection. Sometimes genetic variants become common or rare simply by chance, particularly when populations go through bottlenecks where only a few individuals survive to reproduce. These mechanisms don't operate in isolation. In real populations, mutation generates new variation. Recombination and sexual reproduction shuffle it into new combinations. Natural selection favors certain variants over others. Gene flow moves alleles between populations, and genetic drift causes random fluctuations. The interplay of these processes, operating over countless generations, produces the patterns of evolution we observe. Understanding these mechanisms is crucial because they explain both the gradual nature of evolutionary change and its creativity. Evolution doesn't happen in giant leaps. It proceeds through the accumulation of small changes, each favored by selection or drifting through populations by chance. Given enough time, these small changes can accumulate into transformations so dramatic that descendants bear little resemblance to their ancestors. Single-celled organisms gave rise to multicellular life. Fish-like creatures colonized land. Reptilian ancestors gave rise to birds. And a lineage of tree-dwelling primates eventually produced humans capable of pondering their own origins. As you drift toward sleep tonight, imagine the patient work of evolution, operating generation after generation, shaping life through the simple mathematics of variation, heredity, and differential success. It's a process without consciousness, without intent, yet it has produced the stunning complexity and diversity of the living world. Now, let's turn our attention to one of the most tangible forms of evidence for evolution: the fossil record, those preserved remnants of ancient life that allow us to peer into vanished worlds. Fossils are windows into deep time, preserved traces of organisms that lived millions or even billions of years ago. They form when dead organisms are buried in sediment before they can completely decay. Over time, minerals infiltrate the remains, replacing organic material with stone while preserving the structure. The process is rare. Most organisms die and decompose completely, their organic matter recycled by bacteria and scavengers. Only under special circumstances, when burial happens quickly and conditions are right, does fossilization occur. Despite the rarity of fossilization, paleontologists have recovered millions of fossils representing thousands of extinct species. These fossils are not randomly distributed through Earth's rock layers. Instead, they show a clear pattern. Simpler organisms appear in older rocks, while more complex forms appear later. The deepest, oldest rocks contain no fossils at all, dating to times before life existed. Moving up through the layers, we encounter the first signs of life. Simple, single-celled organisms preserved in rocks that are over 3 billion years old. For billions of years, life remained microscopic and simple. Then, around 600 million years ago, something remarkable happened. Complex, multicellular organisms exploded onto the scene in an event paleontologists call the Cambrian explosion. Within a relatively brief geological period, most major animal body plans appeared in the fossil record. Trilobites scuttled across ancient seafloors. Strange creatures unlike anything alive today built reefs and burrowed through sediment. The oceans teemed with diversity. But what the fossil record shows most powerfully is change over time. The organisms preserved in ancient rocks are not the same as those in younger rocks. Trilobites, which dominated Paleozoic seas for nearly 300 million years, are completely extinct. They have no living descendants. The same is true for countless other groups. Ammonites, which swam in ancient oceans with their coiled shells. Dinosaurs, which ruled terrestrial ecosystems for over 160 million years. Saber-toothed cats, which prowled Ice Age landscapes. All these forms flourished and then vanished, replaced by new forms. The succession of life forms in the fossil record is not random. It shows patterns consistent with evolution. Birds don't appear in rocks older than dinosaurs. Mammals don't appear before reptiles. Flowering plants don't appear before simpler plants. The order is always the same, no matter where on Earth the rocks are found. This consistent ordering makes sense if life has evolved over time, with new groups arising from earlier ones. It would make no sense if all species were created simultaneously, or if their appearance were random. Perhaps the most compelling fossil evidence comes from transitional forms, organisms that possess characteristics of two different groups, providing glimpses of evolutionary transitions. Archaeopteryx, discovered in 1861, just 2 years after Darwin published Origin of Species, is a beautiful example. This creature, which lived about 150 million years ago, has characteristics of both reptiles and birds. It has feathers and wings like a bird, but it also has teeth, a long bony tail, and claws on its wings. Features seen in reptilian dinosaurs, but not in modern birds. Archaeopteryx appears exactly where evolutionary theory predicts. in rocks younger than typical dinosaurs but older than fully modern birds. The fossil record documents the evolution of whales from land-dwelling mammals in exquisite detail. Early whale fossils, dating to around 50 million years ago, show creatures that were still partly terrestrial. Pachycetus had legs adapted for walking on land, but features of the skull that link it to modern whales. Ambulocetus, which lived slightly later, was more aquatic, with shorter legs and a longer, more powerful tail. Its name means walking whale, and it probably lived like a modern crocodile, hunting in shallow coastal waters but still able to move on land. Later, forms like Dorudon were fully aquatic, with vestigial hind limbs so reduced they couldn't have supported the animal's weight on land. Modern whales retain tiny remnants of these hind limbs, buried deep within their bodies, evolutionary echoes of their walking ancestors. Tiktaalik, discovered in the Canadian Arctic in 2004, provides another stunning transitional example. This creature lived about 375 million years ago, during a time when the first tetrapods, four-limbed vertebrates, were making the transition from water to land. Tiktaalik has fins like a fish, complete with fin rays. But hidden inside those fins are bones arranged like the limb bones of tetrapods. A single upper arm bone, two lower arm bones, and a series of smaller bones that in later tetrapods would form wrists and digits. It also has a neck, allowing it to turn its head independently of its body, unlike fish, but like tetrapods. And it has a flat head with eyes on top, suggesting it spent time in shallow water looking upward, perhaps hunting on the water's surface or even venturing briefly onto mudflats. The human fossil record, though it occupied a relatively brief span of time compared to the entire history of life, is remarkably rich. More than 6,000 individual fossils of ancient human relatives have been discovered, representing over 20 different species. These fossils show a clear progression from more ape-like ancestors to modern humans. Australopithecus afarensis, which lived between 3 and 4 million years ago, had a brain about the size of a chimpanzee's but walked upright on two legs, as evidenced by its skeleton and by preserved footprints found in volcanic ash in Tanzania. Later species show gradual increases in brain size, changes in skull shape, reduction in the size of teeth and jaws, and other anatomical shifts. Homo erectus, which appeared about 2 million years ago, had a brain significantly larger than Australopithecus, made sophisticated stone tools, and may have used fire. Neanderthals, our closest extinct relatives, had brains as large as or larger than modern humans and exhibited complex behaviors, including burial of their dead and creation of symbolic objects. These transitional fossils don't represent direct ancestors necessarily. Evolution isn't a ladder leading inevitably to humans or any other modern species. It's more like a branching bush, with many lineages splitting off, most eventually going extinct. The fossils we find represent points on various branches, giving us glimpses of the diversity that existed at different times. But collectively, they demonstrate the reality of evolutionary change. They show organisms with characteristics intermediate between earlier and later forms, appearing at the expected times and in the expected sequence. The incompleteness of the fossil record, which Darwin worried about, is no longer the problem it once seemed. We now understand why fossilization is rare and why we shouldn't expect to find fossils of every organism that ever lived. The remarkable thing is not what's missing, but how much we have found. Every year, new fossils are discovered, filling gaps, answering questions, and occasionally revealing surprises. Each discovery enriches our understanding of life's history, but none contradicts the basic pattern of evolution. Instead, they confirm and refine it. As you settle deeper into rest, imagine the vast parade of life that has marched across this planet. Trilobites in ancient seas, giant ferns in coal swamp forests, dinosaurs beneath Mesozoic skies, Ice Age mammals across tundra. All these forms lived, reproduced, adapted, and eventually went extinct, leaving behind their stony signatures in Earth's crust. We are part of this ongoing story, one more chapter in a book that has been writing itself for billions of years. Next, we'll explore another powerful line of evidence for evolution, one written not in stone, but in the very molecules of life: DNA. If fossils are messages from the past written in stone, DNA is a message written in chemicals, a language that all living things on Earth share. This universality is itself evidence of common ancestry. Every organism, from bacteria to redwood trees to human beings, stores genetic information in DNA molecules using the same four-letter chemical alphabet. The instructions for building and operating a bacterium and the instructions for building and operating a human are written in the same language, using the same code. This would be an extraordinary coincidence if life had multiple independent origins. It makes perfect sense if all life descended from a common ancestor that used this system billions of years ago. DNA, deoxyribonucleic acid, is a molecule shaped like a twisted ladder, the famous double helix. The rungs of the ladder are made from pairs of chemical bases: adenine pairs with thymine, and guanine pairs with cytosine. The sequence of these base pairs along the DNA molecule encodes genetic information. Stretches of DNA, called genes, contain instructions for making proteins, the molecular machines that carry out most cellular functions. The order of bases in a gene determines the order of amino acids in the protein it codes for, which in turn determines the protein's shape and function. When we compare DNA sequences between different species, we find a pattern entirely consistent with evolution. Closely related species have very similar DNA sequences, while distantly related species differ more. This is exactly what we'd expect if species descended from common ancestors, accumulating changes over time. The more recently two species shared a common ancestor, the less time there has been for changes to accumulate, and the more similar their DNA remains. Humans and chimpanzees, our closest living relatives, share approximately 98 to 99% of their DNA sequences. This doesn't mean we're 99% chimpanzee, or that only 1% of our DNA makes us human. The differences, though small in percentage terms, have profound effects. But the overwhelming similarity of the sequences confirms what comparative anatomy and the fossil record suggest. Humans and chimpanzees shared a common ancestor relatively recently in evolutionary time, probably between 5 and 7 million years ago. We can extend these comparisons further. Humans are more similar to other great apes, gorillas and orangutans, than to monkeys, and more similar to monkeys than to other mammals. The pattern continues. Humans are more similar to other mammals than to birds, more similar to birds than to reptiles, more similar to reptiles than to fish, and so on. We can construct a family tree of life based purely on DNA similarities, and that tree matches the relationship suggested by anatomy and the fossil record. The correspondence is remarkable and would be inexplicable without evolution. Molecular biologists can even use DNA to estimate when different species diverged from common ancestors. The idea is based on the observation that mutations accumulate at roughly steady rates over long periods. This allows DNA sequences to function as a molecular clock. By comparing the number of differences in a particular gene between two species and knowing the approximate rate at which mutations occur, scientists can estimate how long ago those species shared a common ancestor. Molecular clocks have their limitations and must be calibrated using fossil evidence, but they've proven remarkably useful for dating evolutionary divergences, especially for organisms with poor fossil records. One of the most compelling pieces of molecular evidence for evolution comes from endogenous retroviruses. Retroviruses are viruses that insert their genetic material into their host's DNA. Occasionally, a retrovirus infects a germ cell, an egg or sperm, and its DNA becomes a permanent part of the host's genome, passed down to all descendants. These integrated viral sequences, called endogenous retroviruses, accumulate mutations over time just like other parts of the genome. Eventually, they become non-functional molecular fossils sitting in our DNA. Humans carry dozens of these endogenous retroviral sequences, remnants of infections that happened to our ancestors millions of years ago. What's remarkable is that we share many of these same sequences with other primates, inserted in exactly the same locations in our genomes. The probability of retroviruses independently inserting themselves in the exact same spots in different species is vanishingly small. The only reasonable explanation is common ancestry. These viral sequences infected an ancestor common to humans and other primates, and we've all inherited them. Chromosome fusion provides another elegant demonstration of human evolution. Humans have 23 pairs of chromosomes, while chimpanzees, gorillas, and orangutans all have 24 pairs. This initially seemed puzzling. How could we have fewer chromosomes than our closest relatives? The answer came from examining human chromosome number 2 in detail. It turns out that human chromosome 2 is the result of an end-to-end fusion of 2 ancestral chromosomes, equivalent to 2 separate chromosomes still found in other great apes. The evidence for this fusion is written clearly in the DNA. Human chromosome 2 has telomeres, the protective caps normally found at chromosome ends, in the middle of the chromosome where the fusion occurred. It also has 2 centromeres, the structures that help chromosomes separate during cell division, though one has been inactivated. The fusion happened after our lineage split from chimpanzees, reducing our chromosome number by one pair. DNA also preserves evidence of our evolutionary history in the form of pseudogenes, genes that have been inactivated by mutations but remain in the genome like broken machinery. Many species have non-functional copies of genes that work perfectly well in other organisms. Humans, for instance, have a broken version of a gene for synthesizing vitamin C. Most mammals can make their own vitamin C using this gene, but humans, along with other primates and guinea pigs, cannot. We've inherited a mutated, non-functional copy of the gene from an ancestor that suffered a mutation disabling it. That ancestor must have gotten enough vitamin C from its diet that losing the ability to synthesize it wasn't fatal. We, descendants, still carry the broken gene, a genetic scar recording an ancient mutation. The pattern holds for countless other genes and genetic features. Closely related species share similar genes, similar gene arrangements, similar regulatory sequences, and even similar mistakes and broken genes. The nested hierarchy of similarities, with species grouped into ever larger categories based on shared features, is precisely what evolutionary theory predicts, and precisely what we find when we examine genomes. Molecular evidence also helps us understand how new features evolve. Gene duplication, where a gene is accidentally copied, creates genetic raw material for evolution to work with. After duplication, one copy can maintain the original function, while the other copy is free to accumulate mutations. Usually, these mutations break the duplicate gene, but occasionally they produce something useful, a protein with a new function or an old function enhanced or modified in beneficial ways. Whole genome duplications, where an organism's entire genome is doubled, have occurred multiple times in evolutionary history, particularly in plants. These events create vast amounts of new genetic material that can be molded by selection into new adaptations. The molecular evidence for evolution is overwhelming because it is written in every cell of every organism. We don't have to rely on rare fossils or particular anatomical structures. We can sequence DNA from any living thing and compare it to any other, revealing relationships, tracing histories, and confirming predictions made from other lines of evidence. The genetic basis of inheritance, mysterious in Darwin's time, is now one of evolution's strongest supports. As your mind grows quieter and sleep draws near, consider the molecular threads connecting all life. The same basic machinery, inherited from ancient ancestors, operates in your cells and in the cells of every other living thing. You carry in your genome the history of your species, the marks of ancient mutations, the scars of viral infections long past, and genes shared with creatures great and small. At the molecular level, kinship with all life is not a metaphor. It's a biochemical fact. Now, let's examine evolution happening in real time, in laboratories and in the wild, processes we can observe directly within human lifetimes. One of the most powerful evidences for evolution is that we can watch it happening. Evolution isn't just ancient history preserved in fossils and DNA. It's an ongoing process, operating all around us. Sometimes at speeds that allow us to observe significant changes within years or even months. The evolution of antibiotic resistance in bacteria provides a stark, medically significant example. When antibiotics were first developed in the mid-20th century, they seemed like miracle drugs, capable of curing infections that had previously been deadly. Penicillin, the first widely used antibiotic, could kill many types of bacteria by interfering with their ability to build cell walls. Initially, it was remarkably effective. But within years of penicillin's introduction, resistant bacteria began appearing. What happened? In any large bacterial population, there is variation. Most bacteria are killed by penicillin, but occasionally, through random mutation, a bacterium arises with a slightly modified enzyme or a change in its cell wall that makes it less vulnerable to the drug. In the presence of penicillin, these resistant bacteria have a huge advantage. Normal bacteria die, but resistant ones survive and reproduce. Within hours, a single resistant bacterium can multiply into millions, all carrying the resistance mutation. If a patient doesn't complete their antibiotic course, or if antibiotics are overused, resistant strains spread. Today, antibiotic-resistant bacteria pose a serious threat to public health. Diseases once easily treated now require multiple drugs or new antibiotics, and bacteria are evolving resistance to those as well. This is natural selection in action, driven by the selective pressure we've imposed through antibiotic use. The same process occurs with pesticide resistance in insects and herbicide resistance in weeds. Each time we apply a pesticide, we create a strong selective pressure. Most pests die, but any individuals with genetic variants conferring resistance survive and reproduce. Their offspring inherit the resistance, and over generations, the population shifts. Farmers must use higher doses or switch to different pesticides, but resistance evolves again. This evolutionary arms race demonstrates both the power of natural selection and the importance of using pesticides judiciously to slow the evolution of resistance. Viral evolution happens even faster because viruses reproduce so rapidly and have such high mutation rates. Influenza viruses evolve so quickly that last year's flu vaccine is often ineffective against this year's strains. The virus's genome changes from one season to the next, altering surface proteins that our immune systems recognize. This is why flu vaccines must be reformulated annually, targeting the strains predicted to circulate in the coming season. HIV, the virus that causes AIDS, evolves rapidly within individual patients. When someone is infected with HIV, the virus replicates constantly, producing billions of copies. Each replication offers opportunities for mutations. Most mutations harm the virus or have no effect, but occasionally one confers resistance to antiviral drugs. This is why HIV treatment typically uses combinations of drugs. If a patient takes only one drug, resistant viruses quickly evolve. Combination therapy works because the probability of a virus evolving resistance to multiple drugs simultaneously is much lower. These examples of evolution happening in real time involve microorganisms, which reproduce quickly and have large population sizes, conditions that speed evolution. But we, can also observe evolutionary changes in organisms with longer generation times. Darwin's finches, those birds on the Galapagos Islands that helped inspire Darwin's theory, have been studied continuously since the 1970s by biologists Peter and Rosemary Grant. The Grants have documented evolution occurring over just a few generations. During a severe drought in 1977, many finches died because food became scarce. The drought disproportionately affected finches with smaller beaks, which couldn't crack the large, tough seeds that were the only food available. Finches with larger, stronger beaks survived better and left more offspring. The offspring inherited their parents' beak sizes, so the average beak size in the population increased. When rains returned and smaller seeds became abundant again, selection favored smaller beaks, and the population's average beak size decreased. Back and forth, generation after generation, the Grants documented how environmental changes drove evolutionary shifts in beak size. The peppered moth in England provides another famous example. Before the Industrial Revolution, most peppered moths were light-colored, well camouflaged against lichen-covered tree bark. Dark-colored moths were rare because they were easily spotted and eaten by birds. As industrialization spread, pollution killed the lichen and darkened tree bark with soot. Now, light-colored moths stood out, while dark moths were camouflaged. The proportion of dark moths increased dramatically. After pollution controls were implemented and trees began recovering their lichen, the light moths became more common again. This shift in moth populations, documented over decades, shows natural selection responding to environmental change. Human activities have driven evolutionary changes in many species. Commercial fishing, which selectively removes larger fish, has caused many fish populations to evolve smaller sizes and earlier reproductive maturity. Trophy hunting, which targets animals with large horns or antlers, has led to decreases in horn and antler size in some populations. Urbanization creates new environments where some species thrive, and we can watch them adapt. Urban bird populations have been shown to evolve different songs than their rural counterparts, likely because urban noise requires adjustments to communication. Laboratory evolution experiments allow scientists to watch evolution under controlled conditions. Populations of bacteria, fruit flies, or yeast can be maintained for hundreds or thousands of generations, with conditions carefully controlled and populations regularly measured. These experiments have shown that populations evolve in predictable ways in response to controlled selective pressures. Bacteria evolve to utilize new food sources. Fruit flies evolve different life history traits depending on whether they're kept in conditions favoring early or late reproduction. The famous long-term evolution experiment with E. coli bacteria, started by Richard Lenski in 1988 and still ongoing, has tracked over 70,000 bacterial generations, documenting the evolution of novel traits, including the ability to metabolize a hydrate, a food source that ancestral E. coli couldn't use. These observations of evolution in action are important because they demonstrate that the mechanisms proposed by evolutionary theory actually work. Evolution isn't just a historical inference. It's a process we can study directly, manipulate experimentally, and even predict. When we understand the selective pressures acting on a population and the genetic variation present, we can often predict how that population will evolve. The speed at which evolution can occur depends on several factors: generation time, population size, mutation rate, strength of selection, and amount of genetic variation. Organisms with short generation times and large populations can evolve more quickly than those with long generation times and small populations. Strong selection speeds evolution, while weak selection slows it. High genetic variation provides more raw material for selection to act on. Understanding evolution in action has practical importance. In medicine, it helps us manage antibiotic resistance and design more effective vaccines. In agriculture, it guides pest management strategies and crop breeding programs. In conservation, it informs efforts to protect endangered species, which may need to evolve to survive in changing environments. Evolution isn't just theoretical. It's a process that affects human health, food production, and the preservation of biodiversity. As you drift toward sleep, think about the constant dance between organisms and their environments. Every generation represents an opportunity for selection to act, for populations to shift, for new adaptations to arise. Evolution never stops. It continues in every population, in every corner of the globe, shaping life in response to ever-changing conditions. Now, let's journey back through deep time, exploring the grand sweep of evolutionary history from life's origins to the present day. The history of life on Earth spans roughly 3.5 billion years, an almost incomprehensible stretch of time. If we compressed this entire history into a single 24-hour day, humans would appear only in the last few seconds before midnight. Most of Earth's history occurred before our species existed, filled with other forms of life, many radically different from anything alive today. Life itself apparently arose relatively quickly after Earth became habitable. The planet formed about 4.5 billion years ago as a ball of molten rock, gradually cooling until a solid crust formed and oceans condensed. For a time, conditions were hellish, with frequent asteroid impacts and volcanic activity. But by about 4 billion years ago, the surface had stabilized enough that liquid water could exist. Within a few 100 million years, life appeared. We don't know exactly how life originated. It's one of the great unsolved questions in science, but we have reasonable hypotheses. The early Earth had simple organic molecules delivered by meteorites and produced by chemical reactions in the atmosphere and oceans. Lightning, ultraviolet radiation, and volcanic heat provided energy to drive reactions that built more complex molecules. At some point, perhaps in tide pools, around deep-sea hydrothermal vents, or in other favorable locations, molecules assembled into structures capable of copying themselves. These first replicators weren't alive in the modern sense. They were probably just strands of RNA or similar molecules that could catalyze their own replication. Over time, these replicating molecules became enclosed in lipid membranes, creating primitive cells. Metabolism evolved, allowing these early organisms to harvest energy from their environment. The genetic code was established, perhaps through evolutionary competition between different coding systems. By about 3.5 billion years ago, recognizable single-celled organisms existed, similar in some ways to modern bacteria. For the next 2 billion years, life remained microscopic and relatively simple. This doesn't mean nothing was happening. Evolution was at work, diversifying life into many forms of bacteria and eventually giving rise to a new domain of life, the archaea. Some bacteria evolved the ability to photosynthesize, using sunlight to convert carbon dioxide and water into organic compounds, releasing oxygen as a waste product. At first, this oxygen was absorbed by iron dissolved in the oceans, forming vast deposits of iron oxide that we mine today. But eventually, oxygen began accumulating in the atmosphere, fundamentally changing the planet. The rise of oxygen was catastrophic for many organisms. Oxygen is reactive and toxic to cells that haven't evolved defenses against it. But some organisms evolved ways to not only tolerate oxygen, but use it, employing oxygen-based respiration to extract far more energy from food than anaerobic processes could provide. This innovation would prove crucial for the evolution of complex life. Around 2 billion years ago, another crucial event occurred: the evolution of eukaryotic cells. Eukaryotes, which include all animals, plants, fungi, and many single-celled organisms, have a different cellular structure from bacteria and archaea. Their cells contain a nucleus housing their DNA and various internal compartments called organelles. The most important organelles are mitochondria, which generate energy, and in plants, chloroplasts, which perform photosynthesis. Mitochondria and chloroplasts were once free-living bacteria that were engulfed by other cells in a process called endosymbiosis. Instead of being digested, these bacteria survived inside their hosts, providing benefits. The bacteria gained protection and nutrients, while the host cell gained the bacteria's metabolic capabilities. Over time, the bacteria became integrated into their hosts, transferring much of their DNA to the host nucleus, until they could no longer survive independently. All eukaryotic cells contain mitochondria, descended from that ancient bacterial merger. Plant cells also contain chloroplasts, descended from photosynthetic bacteria engulfed later. Eukaryotic cells, with their compartmentalized structure and abundant energy from mitochondria, could grow much larger than bacteria and eventually evolved into multicellular organisms. The first multicellular creatures were simple, probably resembling modern filamentous algae. But by about 600 million years ago, complex animals had evolved, including some that left traces in the fossil record. Then, about 540 million years ago, came the Cambrian explosion, a relatively sudden diversification of animal life. Within a few tens of millions of years, a blink of an eye geologically, most major animal body plans appeared. Arthropods, ancestors of modern insects and crustaceans, scuttled across the seafloor. Chordates, the group that includes vertebrates, appeared. Strange creatures, unlike anything alive today, built strange bodies and filled strange niches. The causes of the Cambrian explosion are still debated, but likely factors include rising oxygen levels, the evolution of predation driving evolutionary arms races, and genetic innovations that allowed more complex body plans. After the Cambrian, life continued to diversify and spread. Plants colonized land around 470 million years ago, followed by arthropods and eventually vertebrates. The first land vertebrates were amphibian-like creatures that still depended on water for reproduction. Their descendants gave rise to reptiles, which evolved eggs with protective shells that could be laid on land, freeing them from dependence on water. Reptiles diversified into many forms, including the ancestors of modern turtles, lizards, snakes, and crocodilians, as well as extinct groups like dinosaurs and pterosaurs. Mammals evolved from a group of reptile-like creatures about 200 million years ago. Early mammals were small, probably nocturnal, living in the shadows of the dinosaurs that dominated terrestrial ecosystems. Birds evolved from small theropod dinosaurs around 150 million years ago. The first birds still had many dinosaurian features, including teeth and bony tails, but they had feathers and could fly. Earth's history is punctuated by mass extinctions, events where large percentages of species died out in geologically brief periods. The most famous extinction event occurred 66 million years ago, at the end of the Cretaceous period, when an asteroid impact and massive volcanic eruptions caused environmental catastrophe. The dinosaurs, except for birds, went extinct, along with many other groups. But mass extinctions, though devastating, create opportunities. The extinction that ended the dinosaurs' reign opened up ecological niches that mammals rapidly filled. In the 10 million years following the extinction, mammals diversified from small, nocturnal creatures into a vast array of forms, including marine mammals, flying mammals, and eventually the primates that gave rise to humans. These major transitions in evolution, the origin of life, the evolution of photosynthesis, the rise of eukaryotes, the advent of multicellularity, the colonization of land, the evolution of flight, each opened new possibilities and led to new radiations of diversity. Evolution is not a march of progress toward increasing complexity. Many successful organisms remain simple. Bacteria have thrived for billions of years and show no signs of disappearing. But evolution does explore possibilities, and some of those possibilities involve greater complexity, larger size, more sophisticated behaviors, and eventually consciousness. The history of life reminds us that evolution operates on timescales almost beyond human comprehension. The processes we see operating today, mutation, selection, genetic drift, have been at work for billions of years, gradually shaping and reshaping life. Continental drift has rearranged the geography of the planet. Climates have oscillated between hothouse and icehouse conditions. Oceans have risen and fallen. Through it all, life has persisted, adapted, and diversified, creating the biosphere we inherit today. As you rest, imagine the vast parade of life across deep time. Single cells in ancient seas, strange creatures in Cambrian oceans, plants greening the bare rocks of continents, dinosaurs beneath Mesozoic skies, mammals spreading across a cooling world. Each era had its own characteristic fauna and flora, each ultimately giving way to new forms. And the process continues. with us as both observers and participants. Let's turn now to the specific story of human evolution, tracing our own lineage from primate ancestors to modern Homo sapiens. Human evolution is a topic that touches us personally because it's our own story. Understanding where we came from, what makes us human, and how we're related to other primates helps us understand ourselves. The evidence for human evolution is extensive, drawn from fossils, DNA, comparative anatomy, and archaeology. Humans belong to the order primates, which also includes apes, monkeys, and prosimians like lemurs and tarsiers. Within primates, we're part of the great apes, a group that includes chimpanzees, bonobos, gorillas, and orangutans. Of these, chimpanzees and bonobos are our closest living relatives. We shared a common ancestor with them between 5 and 7 million years ago, relatively recently in evolutionary terms. That common ancestor probably resembled modern chimpanzees in some ways. It likely lived in African forests, was knuckle-walking and tree-climbing, had a brain about the size of a modern chimpanzee's, and lived in social groups. From this ancestor, 2 lineages diverged. One leading to modern chimpanzees and bonobos, the other to humans and our extinct relatives, collectively called hominins. The earliest definitive hominins appear in the fossil record around 6 to 7 million years ago. These early bimes were still quite ape-like, but they show the first hints of bipedalism, walking upright on 2 legs. Bipedalism is the defining characteristic of hominins, the feature that set our lineage apart from other apes. Why bipedalism evolved is debated. It may have offered advantages in spotting predators over tall grass, reaching food in trees, carrying food or infants while moving, or conserving energy during locomotion. Whatever the cause, bipedalism became central to hominin evolution. Australopithecus afarensis, which lived between 3 and 4 million years ago, is one of the best-known early hominins. The most famous Australopithecus fossil is Lucy, a partial skeleton discovered in Ethiopia in 1974. Lucy and her relatives were bipedal but retained features suited for tree climbing. Their arms were relatively long, their fingers curved, and their shoulders oriented like those of apes that spend time in trees. Their brains were small, about the same size as chimpanzee brains. But their legs and pelvis were adapted for walking upright, and fossilized footprints from Laetoli in Tanzania, made by Australopithecus around 3.5 million years ago, show a walking gait similar to modern humans. Several other Australopith species lived in Africa between 4 and 2 million years ago. Some, like Australopithecus africanus, were relatively lightly built. Others, like Paranthropus robustus and Paranthropus boisei, were heavily built with massive jaws and teeth adapted for chewing tough vegetation. These robust Australopiths represent an evolutionary experiment in specialized plant eating. They went extinct around 1 million years ago, leaving no descendants. The genus Homo, to which modern humans belong, appeared around 2 to 3 million years ago. The earliest species, like Homo habilis, had larger brains than Australopiths and made simple stone tools. These tools, flaked stones with sharp edges, are the earliest clear evidence of hominin technology. Tool use probably predates Homo. Chimpanzees use tools, so our common ancestor likely did too. But the systematic manufacture of stone tools represents a cognitive leap. Homo erectus, which appeared about 2 million years ago, was a crucial species in human evolution. Homo erectus had a brain significantly larger than earlier hominins, about 2/3 the size of modern humans. They made more sophisticated tools, including hand axes shaped with multiple flakes, indicating planning and skill. Homo erectus was the first hominin to leave Africa, spreading into Asia and possibly Europe. Fossils have been found in China, Indonesia, and elsewhere. They controlled fire, at least in later populations, which would have provided warmth, protection from predators, and the ability to cook food, making more nutrients available and reducing chewing time. Around 300 to 400,000 years ago, brain size increased again, approaching modern human size. Several species or populations coexisted during this period, including Neanderthals in Europe and Western Asia and Denisovans, known primarily from DNA, in Asia. These archaic humans had large brains, made complex tools, used fire, and exhibited behaviors suggesting symbolic thought. Neanderthals buried their dead, used ochre pigments, and may have created cave art. They survived in Europe for hundreds of thousands of years before going extinct about 40,000 years ago. Modern humans, Homo sapiens, evolved in Africa around 300,000 years ago. Early Homo sapiens had modern-looking skeletons, but their cultures were still relatively simple. Around 60 to 80,000 years ago, something changed. Symbolic artifacts appear more frequently in the archaeological record. Shell beads, ochre pigments, elaborate burials, and eventually cave paintings and carved figurines. Language, if it didn't exist before, certainly existed by this time, enabling the transmission of complex information between individuals and across generations. Modern humans began migrating out of Africa around 60 to 70,000 years ago, eventually spreading across the entire globe. They encountered Neanderthals and Denisovans, and genetic evidence shows they interbred with them. Most modern humans outside Africa carry small amounts of Neanderthal DNA, typically 1 to 2% of their genomes. Some populations, particularly in Oceania, also carry Denisovan DNA. So while Neanderthals and Denisovans went extinct as distinct populations, they live on in fragments within us. The story of human evolution is not a simple linear progression from primitive to advanced. Multiple hominin species often coexisted. Evolution produced many experimental forms, most of which went extinct without descendants. The hominin family tree is bushy, not a ladder. But through this complexity, we can trace general trends: increasing brain size, more sophisticated tools, greater behavioral flexibility, and eventually the emergence of complex language, art, and culture. What makes humans unique isn't just our large brains, though that's important. It's the combination of traits. Bipedalism, freeing our hands for manipulation. Complex language enabling sophisticated communication. Culture allowing accumulation of knowledge across generations. and perhaps cognitive abilities that allow us to reason abstractly, plan far into the future, and create symbolic systems like writing and mathematics. These traits didn't all appear at once. They evolved piecemeal over millions of years, each building on what came before. Human evolution continues today. We haven't stopped evolving just because we have technology and culture. Genetic studies show that human populations have evolved different adaptations to local environments. High-altitude populations in Tibet and the Andes have evolved increased oxygen-carrying capacity in their blood. Populations with long histories of dairy farming have evolved lactose tolerance, allowing adults to digest milk. Populations in malarial regions have elevated frequencies of genes, providing some resistance to the disease, even though some of these genes cause sickle cell anemia when present in 2 copies. As you drift towards sleep, consider your place in this long lineage. You carry within you the legacy of millions of years of evolution, the accumulated survival and reproductive success of countless ancestors stretching back through early humans, through Australopiths, through early primates, through mammalian ancestors, through fish-like creatures in ancient seas, all the way to the first flickering life. You are the current endpoint of an unbroken chain reaching back to the origin of life itself. Let's explore additional lines of evidence for evolution, including biogeography and comparative anatomy, which provide their own compelling testimony to life's history. Biogeography, the study of how organisms are distributed across the planet, provides strong evidence for evolution. The distribution of species makes sense in light of evolutionary history and continental drift, but would be puzzling if species were independently created and placed in their current locations. Oceanic islands provide some of the clearest biogeographical evidence for evolution. Islands that formed volcanically and never connected to continents have distinctive flora and fauna. They lack many groups found on nearby continents, but have unusual radiations of species found nowhere else. The Hawaiian Islands, for instance, have no native land mammals except a bat, no native reptiles, and no native amphibians. But they have spectacular radiations of honey creeper birds, descended from a single ancestral species that colonized the islands, and hundreds of species of fruit flies found nowhere else. This pattern makes sense if we understand that islands are colonized by organisms that can reach them over water. Birds can fly there. Seeds can float on ocean currents or be carried by birds. But most land mammals, reptiles, and amphibians cannot cross large ocean gaps. Once organisms reach an island, they encounter empty niches and evolve to fill them. A single finch species arriving on the Galapagos split into multiple species, each adapted to different food sources. Similarly, the ancestral honey creeper in Hawaii gave rise to species with beaks suited for nectar feeding, seed crushing, insect catching, and other specializations. Continental biogeography also supports evolution. Regions that have been isolated for long periods have distinctive fauna, even when the environments are similar. Australia, isolated for tens of millions of years, is dominated by marsupial mammals, kangaroos, koalas, wombats, and many others. Marsupials give birth to tiny, underdeveloped young that complete their development in a pouch. They're rare in other parts of the world, where placental mammals dominate. Why would Australia have mostly marsupials while other continents have mostly placentals? The answer lies in Earth's geological history. When Australia separated from other land masses, it carried a marsupial-dominated fauna with it. Isolated, these marsupials evolved into many forms, filling ecological roles that placentals fill elsewhere. Australia has marsupial carnivores, marsupial diggers, marsupial gliders, even a recently extinct marsupial that resembled a wolf. Comparative anatomy, the study of anatomical similarities and differences between species, reveals patterns consistent with common ancestry. Homologous structures are anatomical features that share a common evolutionary origin, even if they now serve different functions. The forelimbs of vertebrates provide a classic example. Human arms, bat wings, whale flippers, horse legs, and bird wings all have the same basic bone structure. 1 upper arm bone, 2 lower arm bones, a cluster of wrist bones, and 5 sets of bones forming fingers or digits. The arrangement is the same, even though the limbs serve vastly different functions. This makes sense if all these vertebrates inherited the basic forelimb structure from a common ancestor, and evolution modified it for different purposes. It would be puzzling if each species were independently designed, because why would a designer use the same structure for such different functions when other structures might work better? Vestigial structures provide another line of anatomical evidence. These are features that served important functions in ancestral species but have lost their original function in descendants. They're like evolutionary echoes, remnants of the past. Whales have tiny hip and leg bones buried deep in their bodies, useless for locomotion but clear evidence of their descent from four-legged terrestrial ancestors. Blind cave fish have non-functional eyes beneath their skin, inherited from sighted ancestors but degenerated through disuse in the perpetual darkness of caves. Flightless birds like ostriches and kiwis have wings too small for flight, remnants of flying ancestors. Humans have a tailbone, the coccyx, composed of several fused vertebrae at the base of the spine. All that remains of the tail our ancestors possessed. Some vestigial features are molecular. Humans have many pseudogenes, broken genes that no longer function. The olfactory receptor genes, which detect smells, number about 400 in mice but only about half that in humans, with many of the human versions non-functional. Our ancestors probably had better senses of smell, but as vision became more important and olfaction less so, selection stopped maintaining all these genes, and many accumulated mutations that disabled them. The nested hierarchy of life, evident in both anatomy and DNA, is another powerful piece of evidence. Species can be grouped into ever larger categories based on shared features. All mammals have hair, produce milk, have 3 middle ear bones, and possess other mammalian characteristics. Within mammals, primates share features like forward-facing eyes, grasping hands, and flattened nails instead of claws. Within primates, great apes share even more features. This pattern of nested groups, where each level shares features not found in other groups, is exactly what we'd expect from a branching evolutionary process. it's harder to explain if species were independently created, because why would a creator organize life into this particular pattern? Embryological development provides yet another line of evidence. Vertebrate embryos pass through similar early stages, regardless of whether they'll become fish, amphibians, reptiles, birds, or mammals. All vertebrate embryos have pharyngeal arches, structures that in fish develop into gills, but in land, vertebrates become parts of the jaw, ear, and throat. Human embryos briefly have structures reminiscent of tails. These embryological similarities reflect shared ancestry. Developmental programs are conserved across evolution because changing early development is often lethal. So, evolution tends to modify later stages while leaving early patterns intact. Together, biogeography, comparative anatomy, embryology, and other fields create a converging body of evidence, all pointing to the same conclusion. Life evolved through descent with modification from common ancestors. Each line of evidence alone would be suggestive, but together they're overwhelming. They all tell the same story, a story of life's long history on this planet. Let your thoughts settle as we contemplate these patterns. The distribution of species across continents and islands. The similarities in bone structures between creatures as different as bats and whales. The remnants of ancestral features in modern organisms. All these pieces fit together when we understand evolution, creating a picture of life's interconnectedness. Now, let's explore the various forms of natural selection and related processes that drive evolutionary change. Natural selection is not a single, monolithic process. It comes in different forms, each shaping populations in distinct ways depending on the nature of selection pressures and the traits involved. Directional selection, perhaps the most intuitive form, occurs when one extreme of a trait's variation is favored. If larger body size provides advantages, selection favors larger individuals, and the population's average size increases over generations. If darker coloration provides better camouflage, darker individuals survive better, and the population becomes darker. The evolution of antibiotic resistance, the increase in beak size in Darwin's finches during drought, and the darkening of peppered moths during industrialization all exemplify directional selection. Stabilizing selection, in contrast, favors intermediate values of a trait while selecting against extremes. Birth weight in human infants shows this pattern. Babies of average weight have the highest survival rates. Very small babies face health risks, but so do very large babies who create complications during birth. Selection favors intermediate weights, reducing variation in the population without changing the average. Stabilizing selection tends to maintain the status quo, preventing populations from changing in particular traits. Disruptive selection favors extremes while selecting against intermediates. This can lead to a population splitting into two distinct forms. In some African finches, beak sizes show a bimodal distribution, with individuals having either small beaks suited for soft seeds or large beaks suited for hard seeds, and intermediate beak sizes being less common. Disruptive selection can potentially drive speciation if the two forms become reproductively isolated. Sexual selection, first described by Darwin, is a special form of selection driven by mating success rather than survival. Traits that enhance an individual's ability to acquire mates can spread through populations even if they reduce survival. Peacock tails are the classic example. Their elaborate tail feathers are costly to grow, cumbersome to carry, and make males more conspicuous to predators. But peahens prefer males with large, colorful tails, so these traits spread despite their survival costs. Sexual selection can drive the evolution of extreme traits, explaining many of the dramatic differences between males and females in species where males compete for female attention. Sexual selection operates through two mechanisms: competition between members of the same sex, usually males, for access to mates, and mate choice, where one sex, usually females, chooses mates based on particular traits. Male-male competition leads to evolution of weapons like antlers and horns, large body size, and aggressive behaviors. Mate choice leads to evolution of ornaments, bright colors, elaborate courtship displays, and songs. Why do females often prefer males with elaborate displays? Several hypotheses exist. Bright colors and large ornaments might indicate good health and high-quality genes because only healthy males can afford the costs of producing them. Elaborate displays might demonstrate motor skills, coordination, and lack of parasites. Or preferences might arise through arbitrary aesthetic biases that, once they spread through a population, become self-reinforcing through a runaway process where females prefer showy males because other females prefer showy males, making showy males' offspring more likely to secure mates. Kin selection explains the evolution of altruistic behaviors that benefit relatives, at cost to oneself. Many animals help raise siblings, defend relatives from predators, or share food with kin. These behaviors can evolve because relatives share genes. By helping a sibling survive and reproduce, an individual helps propagate copies of its own genes that exist in the sibling's genome. The closer the relationship, the stronger this effect. This is why altruism is more common among close relatives than distant kin or non-relatives. Group selection, the idea that selection can favor traits benefiting groups even at cost to individuals, is controversial. Groups with many altruists might outcompete groups with fewer altruists, driving altruism to spread. But within groups, selfish individuals have advantages over altruists, so individual selection tends to eliminate altruism. Group selection can operate under special circumstances, particularly when groups are small, isolated, and frequently go extinct and recolonize. But most biologists think individual and kin selection are more important. Evolutionary arms races occur when species exert reciprocal selective pressures on each other. Predators and prey engage in arms races. Predators evolve better hunting abilities. Prey evolve better defenses. Predators evolve counters to those defenses, and so on. Parasites and hosts engage in similar races. Parasites evolve ways to evade host immune systems. Hosts evolve better defenses. Parasites evolve new evasion tactics. These coevolutionary dynamics can drive rapid evolution and lead to complex adaptations. Plants and their pollinators engage in coevolution as well. Flowers evolve traits that attract specific pollinators. shapes that fit particular insect mouth parts, colors visible to pollinator eyes, scents appealing to pollinator preferences. Pollinators, in turn, evolve traits matching flower characteristics. Some relationships become highly specialized, with certain orchids pollinated only by single bee species, and certain yucca plants pollinated only by specific yucca moths. Frequency-dependent selection occurs when the fitness of a trait depends on its frequency in the population. In some species, rare color morphs have advantages because predators form search images for common forms and overlook rare ones. This maintains variation in the population. In certain fish, a left-mouthed morph and a right-mouthed morph coexist because prey develop defenses against the common morph, giving the rare morph an advantage. Artificial selection, practiced by humans for thousands of years, demonstrates selection's power. We've dramatically transformed domesticated species through selective breeding. Dogs descend from wolves, but come in an astonishing variety of sizes, shapes, and temperaments, all produced through human selection for desired traits. Crops like corn, wheat, and rice bear little resemblance to their wild ancestors, having been selected for larger seeds, more edible parts, and lack of natural seed dispersal. Modern breeding techniques accelerate these processes, allowing targeted selection of specific traits. Understanding these different forms of selection helps us appreciate evolution's versatility. Selection can push populations in particular directions, maintain existing states, split populations into multiple forms, or drive reciprocal changes in interacting species. The outcomes depend on the nature of variation, the selective pressures, and ecological contexts. But in all cases, the basic mechanism remains. Individuals with traits that enhance survival and reproduction leave more offspring, and those traits become more common. Let your mind drift over these concepts. Evolution operates through many pathways, shaped by ecological interactions, mating dynamics, and the endless interplay between organisms and their environments. It's a process of exquisite subtlety and profound creativity. Before we conclude, let's address some common misconceptions about evolution, clarifying what the theory does and doesn't claim. Evolution is often misunderstood, leading to confusion and unnecessary controversy. Clearing up these misconceptions helps us appreciate what evolutionary theory actually says. First, evolution is not just a guess or speculation. When scientists call evolution a theory, they're using theory in its scientific sense, meaning a well-substantiated explanation supported by vast amounts of evidence. In science, theories are the highest form of knowledge, comprehensive frameworks that explain and unify many observations. Calling evolution just a theory misunderstands how science works. Gravity is also a theory, but no one doubts that objects fall. Evolution doesn't claim that humans evolved from chimpanzees. This is a common misconception. Humans and chimpanzees both evolved from a common ancestor that lived millions of years ago. That ancestor was neither a modern chimpanzee nor a modern human. Both lineages have evolved since the split. Chimpanzees are our cousins, not our parents. Evolution is not a ladder of progress leading from simple to complex, from primitive to advanced. Evolution doesn't have a direction or goal. It's simply populations adapting to their environments. Bacteria are just as evolved as humans. They've simply evolved differently. Many evolutionary changes involve simplification rather than increased complexity. Parasites often lose organs and systems present in their ancestors. Cave animals lose eyes and pigmentation. These losses are adaptive in environments where the lost features would be costly to maintain without providing benefits. Natural selection is not random. Mutations are random in the sense that they don't arise in response to need. But selection is the opposite of random. It's a non-random process that systematically favors certain variants over others based on their effects on survival and reproduction. The outcome of evolution can be quite predictable when selective pressures are consistent. Evolution doesn't violate the second law of thermodynamics, which states that entropy in closed systems increases over time. Earth is not a closed system. It receives energy from the sun. Living things use this energy to build complexity, just as a growing crystal uses energy from its surroundings to build ordered structures. There's no thermodynamic problem with evolution. Individual organisms don't evolve during their lifetimes in the genetic sense. Populations evolve over generations as gene frequencies change. Individuals can adapt during their lives through learning, acclimation, and physiological adjustments. But these changes aren't evolution unless they're genetically based and heritable. Evolution doesn't require millions of years for all changes. As we've seen with antibiotic resistance and Darwin's finches, significant evolutionary changes can occur within years or decades when selection is strong. The rate of evolution depends on generation time, population size, strength of selection, and available variation. Transitional fossils are not rare. Every fossil is transitional in some sense, occupying a position between ancestors and descendants. But we have many examples of fossils showing intermediate characteristics between major groups. Archaeopteryx between dinosaurs and birds. Tiktaalik between fish and tetrapods. Various fossils showing whale evolution from land mammals, and extensive hominin fossils documenting human evolution. Evolution doesn't claim to explain the origin of life, only how life has changed since it originated. The origin of life is a separate question, studied in the field of abiogenesis. Evolution begins once replicating, heritable molecules exist. How those first replicators arose is an open question, though scientists have proposed and tested various hypotheses. Evolution is not incompatible with the existence of purpose or meaning in life. Science describes how things work, not why they exist or what they mean. Those questions lie outside science's scope. People can accept evolution while maintaining religious beliefs, finding meaning in existence, and living purposeful lives. Many religious traditions have reconciled their teachings with evolutionary science, viewing evolution as the process through which divine creation unfolded. Finally, gaps in the fossil record don't disprove evolution. Fossilization is rare, and we wouldn't expect to find fossils of every organism that ever lived. The remarkable thing is not what's missing, but how much evidence we found. Each new fossil fills gaps and provides more detail, but the overall pattern has been clear for over a century. Life has changed dramatically over time through branching descent with modification. Understanding these clarifications helps us engage with evolutionary science accurately, appreciating its scope, its evidence, and its implications without misconceptions clouding our understanding. As we near the end of our journey, take a moment to appreciate the clarity that comes from understanding. Evolution is not a threat to meaning or purpose. It's simply the best scientific explanation for life's diversity, supported by multiple converging lines of evidence. It changes nothing about our capacity to wonder, to love, to find beauty in existence. If anything, understanding our connection to all life enriches our experience of the world. Now, let's bring our exploration to a gentle close, reflecting on evolution as an ongoing process and the profound insights it offers. We've traveled through deep time tonight, from Darwin's observations on the Galapagos to the molecular evidence written in every cell, from fossils of ancient creatures to evolution happening in real time around us. We've explored the mechanisms that drive change and the patterns that reveal life's interconnected history. Through it all, one truth emerges clearly. Evolution is real, well documented, and fundamental to understanding life. Evolution continues today, all around us and within us. Our species hasn't stopped evolving. Human populations continue to adapt to local environments, to disease pressures, to dietary changes. We're not the finished product of evolution, but participants in an ongoing process. The bacteria in our bodies evolve. The crops we grow evolve. Wild populations respond to climate change, pollution, habitat loss, and other pressures we've created. Understanding evolution helps us predict and manage these changes, from developing new antibiotics to preserving endangered species to adapting agriculture to changing conditions. The beauty of evolution lies in its simplicity and its power. From a few basic principles, variation, heredity, selection, emerges all the complexity of life. There's something profound in realizing that we're connected to every living thing, that we share ancestry with redwood trees and hummingbirds and mushrooms and bacteria. The same genetic code, the same cellular machinery, the same biochemical pathways run through all of us. Inheritance from our common ancestor billions of years ago. Evolution also reveals nature's creativity. Through random mutation and non-random selection, life has explored an almost infinite variety of forms and functions. It's produced sonar in bats, bioluminescence in deep-sea creatures, camouflage in countless animals, photosynthesis in plants, and consciousness in humans. Each lineage has its own story of adaptation and survival, its own unique solution to the challenges of existence. Perhaps most importantly, evolution gives us perspective. Our species is young, having existed for only a few 100,000 years. We're one twig on a vast tree of life, successful for now, but not guaranteed permanence. The fossil record reminds us that most species that have ever lived are now extinct. This should inspire both humility and appreciation. We're part of something much larger than ourselves, a process billions of years old that will continue long after we're gone. Yet we're also unique. Our consciousness, our ability to understand evolution itself, our capacity to shape our future through knowledge and technology. These distinguish us from other species. We have the power to drive species to extinction or to preserve them, to alter climates or to stabilize them, to understand our origins or to remain ignorant. With this power comes responsibility to use our knowledge wisely, to recognize our place in nature, and to act as stewards of the living world we're part of. The theory of evolution stands as one of humanity's greatest intellectual achievements, rivaling the discoveries of gravity, relativity, and quantum mechanics. It unifies biology, explaining patterns that would otherwise be mysterious. It has practical applications in medicine, agriculture, and conservation. And it enriches our understanding of ourselves and our place in nature. As you settle into sleep tonight, consider the journey we've taken. From volcanic islands to ancient rock layers, from DNA molecules to living populations, the evidence for evolution surrounds us. It's written in our bodies, carrying vestiges of our past. Wisdom teeth we no longer need, an appendix whose original function is lost, goosebumps that once raised fur we no longer have. It's written in our DNA, sharing genes with chimpanzees and bacteria alike. It's written in our behavior, revealing deep evolutionary roots of emotion, cooperation, and cognition. You are the product of billions of years of evolution, the current representative of an unbroken lineage stretching back to the beginning of life. Every ancestor in your line successfully survived to reproductive age, passing genes forward through countless generations, through environmental changes and challenges, through ice ages and warm periods, through prehistoric times to the present moment. That's a remarkable achievement, one you share with every living thing on Earth. If this exploration brought you peace, helped you understand, or sparked your curiosity, I invite you to like and subscribe. Your support helps this channel continue bringing science to those seeking gentle knowledge in quiet moments. Share your thoughts in the comments if you'd like. I enjoy reading your reflections and questions. But for now, simply rest. Let these ideas drift through your mind like clouds across a sky. Evolution is patient. Life is persistent. And you are part of this magnificent ongoing story, a story that began billions of years ago and continues through you, through me, through every creature sharing this planet. Breathe deeply. Let your body relax completely. The night is here to hold you. Evolution took billions of years to create this moment, this consciousness, this capacity to wonder and to know. Rest in that knowledge. You've done enough thinking for tonight. The ancient process that shaped all life continues its slow, patient work. And you can simply drift away, knowing you're connected to everything that lives, everything that has ever lived, through the grand, beautiful process of evolution. Good night.